So here's our agenda for our 90-minute session today. First, I'll tell you a bit more about the ASXL Rare Research Endowment Foundation, the resources that we have available for families, and how we help connect the professional community. Then we'll hear from our two expert speakers, Dr. Loren Peña and Dr. Vandana Shashi, both of whom are medical doctors and researchers. They'll present an overview of Shashi-Pena Syndrome and some research updates, and then we'll move to questions from family that include both clinical and research topics. We'll start with the questions that were submitted when you registered for the session, and then we can take some live questions as well. So the ASXL Rare Research Endowment Foundation was founded by a parent in 2018 to focus on research. Today, we're led by a team of volunteers who are family members, doctors, and researchers, a highly motivated team of people who are directly impacted by ASXL disorders. At the end of today's session, and in a few minutes, I'll share a few ways that you can get involved with us. So our goal as an organization is to advance our understanding of ASXL genes, how they work and what they do, and how to improve the care of people with ASXL syndromes. Our mission encompasses the study of 3 ASXL syndromes: ASXL1, Boring-Opitz syndrome, ASXL2, Shashi-Pena syndrome, and ASXL3, Bainbridge-Roper syndrome. These genes and syndromes are unique, but they have a lot in common. What we learn about one of them can benefit our universal knowledge of the other two. The rising tide of knowledge benefits everyone affected by an ASXL disorder. We are excited and hopeful to return to an in-person ASXL Family Conference next year in July 2022 in Los Angeles, California. This conference is a great opportunity to meet other families, talk to researchers, and learn from the world's foremost clinical experts. We will announce the specific date when it's been confirmed, and we hope to see you in California next year. We also offer a resource library on our website for families that includes recordings of past sessions, recordings of past conferences and articles. The resource library is where the recording of today's session will live. One of our strategic priorities for this year is to build that library, so please stay tuned for future sessions with medical professionals and researchers. And in fact, I'm pleased to announce today that a new webinar will be available in September with Dr. Rosanna Wexford, who is a clinician scientist at the Hospital for Sick Kids in Toronto. She received an R Foundation research grant to study genetic aspects of ASXL1 and ASXL3. Dr. Wexford will share an update on her research and then take questions from families on Thursday, September 23rd at 12 p.m. US Eastern Time. This is a session that's intended for families, so her presentation will be in simplified terms that you can understand even if you're not a scientist or doctor yourself. The last resource that I'll mention is not managed by the R Foundation, but it's too important not to share with you in case you haven't found it yet. The Shashi-Pena Syndrome ASXL2 Families support group on Facebook is a wonderful source of connection and information about life with Shashi-Pena Syndrome. One of the first families to receive a diagnosis created this grassroots support group, and there are currently just over 50 members in the group. We also serve as a point of connection for the medical and research community, both to each other and to families. We host the ASXL Professional Network, a growing network of doctors, researchers, and other healthcare providers who treat individuals with ASXL syndromes. Our goal is to help these professionals talk to and learn from each other. We help fund the ASXL Patient Registry, which is like a library of data and biological samples for families with an ASXL disorder. The registry is hosted at the University of California, Los Angeles, and data is available for free to any researcher who would like to use it. The R Foundation encourages families to join the registry to give researchers the information and biological samples they need. And I'll say a little bit more about this in a few minutes. We also fund research grants to advance the study of ASXL disorders. 5 scientists from around the world are currently studying an ASXL syndrome with grants that we have provided. Our research commitments total $550,000 to date, and you can read more about these studies in progress on our website if you'd like to learn more. We also host the annual ASXL Research Symposium to provide doctors and researchers with an opportunity to speak directly to each other, to exchange ideas and develop collaborations. This year's symposium will be held virtually in November, and we look forward to an in-person symposium held in tandem with the ASXL Family Conference next July in Los Angeles. The in-person events are so special because scientists get to meet families who live what they study. Many scientists report feeling a renewed energy to continue their study after meeting families. I mentioned the ASXL Patient Registry. There's strong research in collecting samples and data from families with Shashi-Pena syndrome. You can learn more about the registry on our website, and the URL is on the screen there, or by emailing the research coordinator at UCLA directly, and the email address is also on the screen. The registry includes a questionnaire, and there's also a biobank to collect blood or skin samples, and families from anywhere in the world are eligible and able to join the registry. We're eager to welcome your involvement with the R Foundation. We're funded entirely by families and organizations with a connection to an ASXL syndrome. When you make a donation to the R Foundation, you're helping to advance the understanding of ASXL syndromes and support families through education. There are a couple of easy ways that you can contribute. You can host a Facebook fundraiser, which is really simple and easy to do. You can do that directly from the R Foundation page, Facebook page. If you shop on Amazon, you can select us as your beneficiary of the Amazon Smile program. And you can also include us in your family's charitable giving by making a donation directly. You can make a one-time gift, and we also have a growing monthly donation program called the Sustainer Circle. We also rely on volunteers to bring our programs and operations to life. We're seeking volunteers to help supplement our professional services, including IT infrastructure and support, legal advice, accounting, and communications. We're also looking for program volunteers to help run our research grant program, plan our conference, and build our calendar of educational programming for families. You can visit the Get Involved section of our website to learn more, or you can email me anytime at amanda@rfoundation.org. All right, so now we're going to do a quick audience poll here just to see who's joining us today. So I'm going to launch a poll, and this includes two questions. One is, how old is your loved one with Shashi-Pena syndrome? So if you're a family member, you can answer these. And the second one is, how long has your loved one had the diagnosis of Shashi-Pena syndrome? Right. Okay, I think that's about everybody here. So I'm gonna end the poll. So most everyone here is in the 2 to 5 year range, a couple folks in the 5 to 18-year-old range. And then length of diagnosis, primarily 1 to 2 years and longer than 5 years. And several folks who are saying it's not applicable. So these are some of our observers and clinical and research folks who are on the line with us. All right. So now I'm going to introduce our two speakers today. First, I'll introduce Loren Peña, who is a clinical geneticist. She is an associate professor in the Department of Pediatrics at the University of Cincinnati. Dr. Peña developed the post-exome clinic at the Cincinnati Children's Hospital in an effort to help more children get diagnosed for rare diseases. Dr. Peña is passionate about helping the pediatric community and their families find a diagnosis and develop treatments for genetic disorders. Vandana Shashi is a professor in pediatrics at Duke University. She is a pediatrician and medical genetics specialist. Dr. Shashi has a passion for undiagnosed and rare genetic diseases and helping the families who are impacted by these diagnoses. In addition to her work at Duke, she is an investigator with the NIH for undiagnosed diseases. As you might have guessed, these two experts are the people for whom the syndrome is named. Their work was instrumental in identifying the novel clinical phenotype associated with a variation in the ASXL2 gene. Their work started with a patient that Dr. Shashi saw at the age of 7 with an unidentified genetic condition. With the support of the Undiagnosed Diseases Network, Dr. Shashi and Dr. Peña, who are then colleagues at Duke, searched through other genetic sequencing databases to try to find other cases. Their search led to 5 other children who presented with the same genetic mutation and physical characteristics as the original patient. This discovery classified the symptoms as a unique syndrome that became known as Shashi-Pena syndrome. And I'll turn it over now to Dr. Shashi, who is going to get us started with our overview and updates. So thank you, Dr. Shashi.
Thank you, Amanda, and welcome to everybody who's joined. It's very nice to be able to spend this morning with families. Let me scroll up to the top. Oh, I apologize. I don't know what happened. Okay. So Loren and I are going to do this as a tag team. I will do the initial part and then she will take over and give you more information on how the ASXL syndrome is caused in terms of what do we know so far about how the gene deletion itself causes symptoms. We don't know a lot, but she'll give you an update, and there'll be some future steps that can be discussed. But I'll go through the clinical manifestations a little bit with you first. So as Amanda mentioned, and as many of you know, this syndrome was first described in 2016 in an article we published in the American Journal of Human Genetics. And we had 6 patients, all of whom shared certain features in common with one another. The first one was that the head size was a little larger than typical, and they had certain facial features they shared with one another, and we'll go over that in just a minute. And many of you are familiar with that, so I don't need to spend a lot of time on it. Some children had minor congenital heart disease, like an opening between the upper chambers of the heart, for example. Some children presented with feeding difficulties as a newborn. Many of the children had low muscle tone, and developmental delays and intellectual disabilities were common, ranging from mild to more significant disabilities. So about probably over half the children had seizures, enough to make a diagnosis of epilepsy. Many of the children where we had imaging also had findings suggestive of atrophy of the brain. And there were a few other features that were not seen in most of the children, but enough that we felt like they were probably features of the syndrome. And that is some capillary malformations or birthmarks, red birthmarks on the skin other than on the forehead. Some findings of skeletal abnormalities such as low mineralization of the bones, and sometimes low blood sugars as well. So this is the picture from our publication that shows the 6 children. Actually, one of them was an adult in her 30s. That's the last patient, but this gives you kind of an overview of the facial features. The eyes are slightly set apart, tend to be a little prominent. There tends to be a birthmark in the middle of the forehead, and a few other facial features. And to the right are the brain MRI pictures of 4 of the patients, showing that the brain looks a little smaller. And since then, we've Dr. Peña and I have been very actively trying to kind of be involved with any families or clinicians who reach out to us saying they have a new patient diagnosed with the syndrome. And so putting all that information together, we've come across at least 21 patients that have reached out to us or their clinicians saying that, I think my child or my patient has this disorder, can you speak with us? And so we always reach out to the families as well. But if you kind of break this 21 patients down, what's clear is 11 of those 21 patients have the kind of gene changes involving ASXL2 that we think actually are definitely contributing to the disorder. So for example, where there's a deletion in the last 2 exons of this gene, and Loren will show you a cartoon to kind of explain that in just a few minutes. These kinds of variants where part of the gene is lost are called as loss of function variants. And clearly loss of function of ASXL2, or meaning at least deletion of certain parts of ASXL2, or a kind of mutation that causes the protein not to be as long as it is normally made, those definitely associated with the disorder. So we have 11 of those. And then we have a number of variants where they're called as missense variants. And those are variants where you just have substitution of one DNA letter with another. We are not sure at this point if that causes the typical Shashi-Pena syndrome or it's associated with a slightly different form of the disorder. That still remains to be seen. But if you take the 11 patients that we know of who had these loss of function variants, they had features very similar to those 6 patients that were reported in our paper. And we don't need to go over the individual features. We are all familiar with that. But there were some outlying features in these 11 patients that make us wonder, okay, is the phenotype, meaning the spectrum of differences we see with Shashi-Pena syndrome, is that broader than what we first described in the paper? And some of those features we found in these other 11 patients is there was one patient with an immunodeficiency causing very significant infections. There were 2 patients, including one reported by us, where their QT interval on the ECG was higher, and that can cause some predisposition to arrhythmias. There were 2 children reported with hearing loss, and 3 children with hypothyroidism. I apologize, my dog is outside barking. If anybody can hear, I'm sorry. And there was one patient where the optic nerve was really small. So at this point, we don't know if these are enough for us to say they're part of the condition, but this kind of tells us we need to monitor the literature as well as patient data more to see if these indeed should be included as part of the Shashi-Pena syndrome. And one patient was actually was reported to have only one kidney at birth. Again, two small numbers at this time to know for sure. And then if you look at the patients we know of with missense variants, those four patients, there's a lot of variability between them. They tend not to have the typical facial features or some of those other typical features we see with Shashi-Pena syndrome. And they present with other manifestations we haven't really seen, including increased hunger. A child had tongue fasciculations, where that means there's a neuronal abnormality causing that, absent reflexes, and the intellectual disability was milder in those children. And they had a range of other abnormalities. We don't need to go through these in detail that we are not sure is part of this condition. And one child had a second diagnosis as well of a different syndrome, so we don't know how that's impacting this. And as I said, they tend not to have the facial features. And so for some of these missense variants, we don't know what the parents have. So for example, was that missense variant inherited from a parent who's healthy themselves? Whereas most of the ASXL2 loss of function variants that we know of started off as a new change without either parent having it, meaning they're de novos. In addition, to give you kind of a assessment of what the missense variant patients can look like, there was one of those reported in a recent article on the ASXL2, ASXL1, 2, and 3 disorders. They had one ASXL patient here who had a missense variant, and that was a 13-year-old. And in his case, it was de novo, which lends a little bit more weight to this maybe could be related to Shashi-Pena syndrome than not. This individual presented with some of the features we tend to see in this condition, but there were also other features that did not match, had intellectual impairments that are probably similar to what we see, and had some feeding difficulties, but also had some other features like liver involvement, chronic pancreatitis, we tend not to see. And this child also had an inherited myopathy that was running in the family, so we're not sure how much that's contributing. And here you can see his pictures that were published, and he doesn't have the typical facial appearance that we tend to see. So this tells us that the missense variants are probably in a slightly different category than the loss of function variants, and we need to accumulate more information for both of these categories of variants to kind of characterize the condition better, and to know if there are differences between the missenses and the loss of function variants. We also then went and looked at a disease database called ClinVar, where all the laboratories who do testing such as exomes, not all, most of them report variants in different genes into this database, and they classify them as being whether they're pathogenic or of uncertain significance or benign, etc. And there were a number of variants for ASXL2. As you can see, there were 9 frame shifts and 5 nonsenses. So these are the loss of function ones. It's a little higher than the 11 we had. We think there's overlap between what we know and what was reported here, because the same clinicians who called us, the lab that did that testing is probably putting that variant into this database. But again, to tell you that there is more and more information accumulating about is the Shashi-Pena syndrome over time. So for example, we know only of 4 missenses, and there are 34 missenses reported here, some of which are probably benign. But over time, we'll have much more information that we can have an understanding of how broad the spectrum of differences are with this disorder, how the loss of functions might vary from the missense variants. And then we also speak with you families. Talking to you gives us so much more information than any literature review could. So the last Zoom call we had was in summer of 2020. 8 families joined the Zoom call. And what we found that you as parents and family members express as being most significant are the developmental delays being a concern, and difficulties with communication and behavioral problems. There was one child who still has difficulty maintaining his blood sugar despite being much older and is requiring a feeding tube for this. So this can have, so that made us understand that the features can vary from one child to another, that sometimes low blood sugars can continue to persist beyond the newborn period. And many children show autistic behaviors and sensory integration problems that can impact quality of life for themselves and their families. And of the 8 who attended that Zoom call, 2 children had seizures. And so again, that can have an impact on the child and the family, of course. And 5 children were reported to have scoliosis. So this is kind of a very brief poll, but it kind of gave us the, an understanding of the impact on the families and the children. And with this, I'll turn it over to Loren. I'll stop sharing my screen so she can share the presentation and drive it.
Let me share my screen. Okay, everybody, thank you for the opportunity to talk to the group, and I will take up where Vandana left off. So whenever we tie a gene to a new disorder, it's frequently based on whether there's overlap in the features and if there's enough of what I call the bioinformatic signature that many changes in this gene can be deleterious to its function. And so we did that for ASXL2, but we're still left with the question of what is the function of this gene? And so sometimes we can glean some information regarding function just looking at the protein and for example, specificities in the sequencing that can tell us that there's similarity to other described functions and that we can start thinking about functions for ASXL2 in specific manners. And so what we know is that in fruit flies, Drosophila, there's a homolog that's called ASX, and it's involved in body patterning. So what becomes the thorax, what becomes the portions of the thorax that give rise to the wings, et cetera, et cetera. And this is all based on the notion that we all have a common ancestor. And if we go back to, the primordial, the first organism, the further back you can go, then the more important or more fundamental function this gene might have. So if we take it back to fruit flies, we can see that at least there's some overlap with the ASX gene. Now, there is a conserved domain structure for all the ASXL genes. And so the cartoon at the bottom in gray gives you an idea of what the overlap is between the 3 genes. And so there is an ASXN domain, and that domain is known to participate in binding of DNA. There is an ASXH domain that is an important domain for binding to epigenetic regulators, and I'm going to define that a little bit in a couple of slides. There is an AXM1 and ASXM2 domain, and those are important for interactions between the ASXL2 or the ASXL1 and 3, depending on which one we're talking about. Between that gene product and other proteins. And then down at the end, there's a PhD domain that's important for histone or DNA binding. And so this cartoon here is important for understanding what's frequently talked about as the dogma of gene expression. And so we have our genetic material encoded as DNA, and that is the basis for the testing that we do in the genetics clinic, that we look at specific genes. And that's when we talk about mutations in the gene, what we're saying is that there are changes at the level of DNA that can be important for the downstream function. And so from DNA, we have transcription to an RNA molecule. And then from RNA, we have translation into protein. And so when I talk about protein-protein interactions, what I'm saying is you take the ASXL2 gene, it gets transcribed into RNA and translated into the ASXL2 protein. And then that protein is going to be different and the function is going to be different if there's a mutation that we expect is going to cause disease. And so the question then becomes, what is the problem that mutation brings at the protein level? And so what we think and what the evidence is showing is that the ASXL disorders participate in epigenetic modification. And what that means is that you have DNA, RNA, and protein. And so even epigenetic modifications have nothing to do with DNA. So there's no change at the DNA level, but the change is at the protein level. They regulate how accessible this DNA is to transcription, so becoming RNA. And so in many ways, they can be thought of as on-off switches. So on being gene expression and off being no gene expression. They can also mean off at a specific time or in a specific tissue, and conversely on at a specific time in a specific tissue. And so some of the epigenetic modifications can be the epigenetic modifications are additions of a specific compound in these proteins called histones that bind to DNA, and that compound can be different. And so it can be an acetyl group, a methyl group, phosphorylation, or ubiquination. And so there's even nuance within the scope of epigenetic modifications. And so we also learn a little bit about the tissue specificity and also the downstream regulation effect of the ASXL proteins when we look at the publications of their role in malignancies. And so I have to clarify that this slide doesn't mean that there's a greater risk of malignancy for the ASXL disorders. What I'm saying is that when one has some sort of malignancy, a big window into what is happening at the tissue level might be what genes are activated or inactivated in that tumor. And those can be acquired changes. So the person is not born with them, but might acquire them as the tissue changes on the road to becoming malignant or at the malignant stage. And so all three genes have been described in the context of somatic mutations or changes at the tissue level, not at the egg or sperm level. And for ASXL2 in particular, acute myelocytic leukemia with a specific change in chromosomes there's a greater amount of expression in that specific context, and it's felt like ASXL2 sort of drives this type of blood cancer by affecting how the cells in the bone marrow are activated. There's also some other malignancies in which ASXL2 can be mutated. And the same story applies to ASXL1 and ASXL3, and I've listed there on the slide the types of cancers that have had these changes. It doesn't, this information, I'll underline again, doesn't mean that there's a greater predisposition of cancer in the ASXL disorders. It's just as we're trying to gather data from the published literature about what some of the roles of these proteins might be, we frequently look at the cancer literature as well. And so then we try to learn also what the function might be from knockout experiments or animal models. And knockout experiments are sort of, in a very crude manner, it's sort of like, just taking a bat to a, just taking a, big, large effect size kind of experiment to see if I knock out the genes in a particular model organism, what's going to happen. And so the first question is, are they born? Are they not born? If they are not born, at what stage? How far do they make it in embryonic development? And so for ASXL1 and 2, there is data from mice. And for ASXL2 in particular, it seems like the problem is at the embryonic stage. And we do see that pups are born when you knock out the ASXL2 gene. However, you expect that a litter will be, you will have a particular number of pups, and what we see is that fewer pups are born. So we think that that means that it's, in many ways, it's a very important gene during embryonic development, because we don't see as many pups as we expect to see. We do see that the pups are smaller also. They might have congenital birth defects, and so anterior and posterior segmental anomalies. And that goes back to what I mentioned in the fruit flies too, that this gene is important for patterning in fruit flies. And so we see that too in the ASXL2 knockouts. And there can be also effects in the heart with dilated cardiomyopathy and fibrosis. So there might not be a straight one-to-one relationship between the physical findings in mice to what we see in humans, but this kind of experiment is very helpful and very informative in telling us that the gene is important in embryonic development. And this is more information telling us that we're on the right track here in thinking that changes that are deleterious to the ASXL2 protein could be expected to have some sort of downstream effect in those individuals who have the change. And so there is more information coming out in support of the role of epigenetic modifications in neurodevelopmental disorders. And so this goes back to the idea that we think that the ASXL proteins are important for epigenetic modifications. And then there's the question of, well, what happens if you lose the function of a gene and a protein that is important for some sort of epigenetic modification? And so what the data supports right now is that it looks like the ASXL proteins are part of a protein complex, meaning they don't have a function in and of themselves, but by the protein domains that I talked about in ASXL2 that we predict from the domains that the protein has, then we expect that there's going to be specific binding partners for ASXL2, and then the function is going to be mediated by those complexes for which ASXL2 is one of the proteins that's in the complex. And so we think that this is going to be important. There's information now that there's a growing list of neurodevelopmental disorders in which the fundamental problem is with a problem with epigenetic modifications. The findings in patients can be highly different, and that is just a function of what the binding partners are, what the downstream effects are, in which tissue the protein is supposed to be functional, and at what time in development the protein is going to be functional. And so that's why we think that there's such a wide variation in the clinical findings that we see in the patients, because this family of disease, just by virtue of the wide variety of binding partners that and the tissue specificity that protein might have, that there's just going to be any kind of effect downstream. And so that's the information that we have, and we do think that there's enough information to state that ASXLs are important for neurological development and for embryonic development as well. And we have 3 genes, and there is some phenotype, there is some overlap in the clinical findings, although there is some differences too. And so ASXL2 in particular gives rise to clinical findings that are a little different from boring opiates and Bainbridge ropers caused by ASXL1 and ASXL3. And so in particular, one consistent finding for Shashi-Pena syndrome is that these patients pretty consistently have head sizes that are above what's expected for age. Sometimes their height and their weight might be higher than expected for age as well. They do seem to have a pretty consistent finding, particularly early in life, with white set eyes, with hooded eyelids. And then there's still overlap between all 3. And so this is a slide from the recent paper by Dr. Kudapa, in which there was a comparison of a cohort of patients with the 3 disorders. And so this is a Venn diagram of what is different and what is the same for all 3. And so in the middle, we have the arched eyebrows, the wide set eyes, feeding difficulties, low muscle tone, and cognitive disabilities for all. And then there's some overlap between boring opiates and Shashi-Pena in that some patients will have seizures or seizure disorder, and they have this skin finding of the nevus flammeus, the reddish skin finding in the forehead. And then there are some specific findings between ASXL1 and 3 that can be overlapping. And so you can see how all 3 disorders might have common functions and different functions, and this might at least in part account for the overlap and the differences between all 3. And so then what do we know and what is it that we don't know? We know that all 3 are involved in neurodevelopmental disorders. What we don't know is, I've talked a lot about binding partners, so protein-protein interactions for the ASXL genes. We don't know what the binding partners are for these genes, although we can start to guess. We don't know what the proposed disease mechanism is for the germline variants. And so we think that in genetics terms, that the mechanism of disease supports a gain of function mechanism. But this is an observation that has not been consistently borne out by the research. And so there could be a couple of mechanisms. So we need to understand that a little bit more. And that will tie into therapeutics as well, because that's really at the heart of how we treat genetic disease. We need in vivo functional studies of ASXL2, I'm sorry, 3 as well, but 2, and that will be a plugin that I put in for research too. And then there are some published cases of malignancies in the ASXL disorders, particularly medulloblastoma and nephroblastoma. And so we need to understand the natural history of the conditions a little bit better, and that will allow us to be able to take care of affected individuals by knowing, for example, if there's a predisposition to malignancies. And so for the next step, so we need to understand the natural history of Shashi-Pena syndrome better. And we have a registry for all 3 genes, and there's always recruitment for Shashi-Pena syndrome, because the more information we have, the better our understanding is going to be across the age spectrum, what the complications can be. The families of affected individuals have been very, very generous with their time when we request additional information, because that's where a lot of the knowledge is within the families. We can only access what's in the published literature, but the families are sharing information at all times, and that can be incredibly valuable to the clinicians. We need this longitudinal follow-up that ties into the natural history. And then we need to continue to foster registry participation, and not only sharing clinical data, but also sharing samples. And so there was a chat from there was a message from Dr. Russell here, and an upcoming talk from Dr. Wexberg, who's an expert in epigenetic modification. And so we need to understand if there is an epigenetic signature for variants in ASXL2. And so this goes back to what Dr. Shashi was mentioning, that even though the main types of mutations that have been seen in individuals with classic findings of Shashi-Pena are frame shifts and nonsense variants, we frequently get questions about these missense variants, which are misspellings. And it is hard, even though the findings are not classic for what we know to be Shashi-Pena syndrome, we're unable to answer the question whether these variants can have some sort of downstream effects for cognitive function, for development, et cetera, et cetera. And so having samples to develop a functional assay for all types of variants in ASXL2, and then answer the question of, yes, there is a downstream effect for some of the missense variants, or no, there isn't, then that will help us help families too whenever these questions arise. So participation is huge for both submission of tissue, but the clinical data as well. And so with that, we'd like to acknowledge the Duke Columbia Undiagnosed Diseases Network team, who was instrumental in our publication from 2016, and then also the R Foundation, who has been really helpful in getting families and investigators together so we can all as a group leverage our strong points to understand the conditions. And so if you have any questions, I posted our email addresses here. There's also a closed Facebook group, and if you search for Shashi-Pena syndrome, you can find it and request to be included in the group. You can contact any of us also if you're interested in participating in the registry. So thank you very much for your time.
Wonderful. Thank you to you both. So we're going to move to questions now. And I think a good question to start with are just some of the genetic basis for the syndrome. And so one of the questions that came through was, is the variation or change to the gene caused by anything that the mother did or did not do?
That's easy to answer, no. Excuse me, what happens during, so our genetic, the functions of our genetic machinery are not 100% infallible. And so there's always some sort of error rate at conception or shortly after conception, but usually at conception or with the egg and the sperm. And so we all have some sort of errors, and frequently we don't know what they are because they don't impact our health, but sometimes there's an error that will impact development and will lead to birth defects and differences in appearance and development and all that, and ASXL2 happens to be one of those. But we know that that is not subject to food or drink or medications or activities, so no.
And I would add to what Loren said to say that, we know that 2 to 3% of pregnancies, there can be a spontaneous error of some sort that causes the baby to be born with a genetic difference, whether it's ASXL2 or some other gene that's involved. So these are just random accidents that just happen that are out of the control of any of us.
Thank you. And then along the lines of sort of continuing with the genetic questions here, someone asked just for some clarification on what you were speaking about, Dr. Peña, with some of the connections to some of the cancers. So the question was, are children with Shashi-Pena syndrome more likely to get leukemia? If you could offer some clarification there from your previous presentation.
So we have not heard of anybody at this point. That doesn't really help us say categorically yes or no, and that's just another plug for the registry and understanding the longitudinal history of the condition. So at this point, we have not heard of anybody, but we also know that there are some children that we don't know.
Thank you. Next question, if my child hasn't been diagnosed with immunodeficiency but tends to get sick often, should I have them tested for this and how?
You want to take that, Vandana?
Sure. So I guess I wanted to ask a clarifying question in return. So are you saying your child with Shashi-Pena syndrome is getting sick often? Is that the question?
Yes, I believe so. They haven't been diagnosed with immunodeficiency, but have Shashi-Pena syndrome.
Okay, good. So we just know of one patient so far, and their clinician reached out to us saying that her patient had Shashi-Pena syndrome and also had an immunodeficiency. And talking to the clinician and going function test, it was clear that the difference in immune abnormalities that this child had was probably likely because there was another gene involved that was causing the immunodeficiency. So we've been in communication with that clinician to kind of look at those other immune function related genes, and we haven't heard back as to what they found in that. But in general, of all the other patients that we know that have the frame shifts and nonsense gene variants, we haven't seen specific immunodeficiency. We've seen children who are prone to getting more infections and might fall sick a lot, but that's probably a function of having, say, low muscle tone, for example, or feeding difficulties, aspiration, those kinds of things cause infections rather than a primary immunodeficiency. So the short answer would be is, I doubt that there's an underlying specific immunodeficiency. I would speak with your pediatrician and your geneticist and let them know there's been one case described. We don't know that it's related to the disorder. And if they feel like there aren't any other factors that are causing your child's infections, then it might be reasonable to look for an immune deficiency.
Thank you. All right, next we're going to go to a question about teeth. This is one of the pre-submitted questions. So the question is, my son is 4 and has lost 6 baby teeth, and those are his front, top, and bottom teeth, and then he's grown his adult back teeth. Is this common?
I can take that, Loren. You want to do this one?
No, go ahead.
Okay, and you can jump in if you have other information. We don't know specifically what the teeth are like with this disorder. What our first index patient, for example, did have some dental abnormalities in the sense that when they did x-rays, he actually had very dense bones in his jaws. On the other hand, we've seen other patients reported where their bone mineralization was lower than normal, highly expressed within the bone, especially in the developing teeth. So it's nice to think there could be some dental issues. We just don't know yet. One of the questions I would ask your dentist is to ask them to look at the x-rays and see if there's any abnormalities in the density of the mineralization of the jaw itself, whether it's upper or lower, that could have contributed to some of this premature loss.
Thank you. Next question comes from another clinician. So I've heard and seen that some children with Shashi-Pena syndrome are associated with microcephaly, so small head, rather than macrocephaly, big head. Is this the case? And if so, does this characteristic associate with specific DNA variants?
Go ahead, Loren.
I mean, it's our initial cohort, all except one, if I recall, Vandana had a head circumference that was above the 95th percentile, and the other one had relative macrocephaly at 80th, if I recall, a little bit higher. So the macrocephaly, large head, seems to be a pretty consistent finding, at least in the initial 6 that that we described. I don't think that we have heard of anybody with microcephaly.
I agree. Even the other 11 patients that we know have loss of function variants that have the typical features of Shashi-Pena, they all have microcephaly in our gathering of information. Now, there are some missense variant patients where microcephaly is being described. But again, as for many different reasons, as Loren went over, whether it's clinical or functional, it's hard at this time to know which of those missense variants are actually causing symptoms. So.
Thank you. All right, next we have two questions actually, so I'll combine them here about cord blood. So the question generally is, my child's sibling has cord blood stem cells stored. Is there any information on this providing benefits to individuals with these conditions? And then the second question to tack on to that is, has there been any research regarding cord blood treatment with ASXL patients?
Go ahead, Vandana.
I was just going to say that, cord blood treatments are being used for many different disorders, so I can understand where you're coming from. There is no literature that we know of that has looked at this in Shashi-Pena Syndrome. And we personally don't think that that's definitely a likely possibility in the future as a way of treating it for many different reasons. And if you have a sibling, for example, where you have a cord blood treatment, then there are issues about how much, there are side effects just related to that. Unless it's a patient's own stem cells you're transplanting, there are going to be problems with another individual, even if genetically related, transfusing that. Then you have to have engraftment of those cells into the tissue of interest, such as the brain and other organs. And there's also a developmental time point because many of these changes we see would have occurred really earlier on during embryonic and fetal development. And you always have to ask how much of those can we reverse or change. So I think at this point, it's probably not the most promising way to treat it. Loren, you can add to that.
I mean, I agree with everything that you said. I think that Amanda, I think that there's a question about CRISPR and gene editing in that way, so I can wrap in that answer too. So I think it's very important to understand what the function of these genes are, because there could be multiple approaches to treating these disorders based on what the protein actually does. And so I think the first step is, and it's just a very simple way to understand, at least, if there's downstream effects of these changes in the functional assays for which we are trying to gather samples. So that will be an easy yes or no, I see downstream effects, or no, I do not. And so if we do see them, that could be a functional assay that's a helpful assay. CRISPR, I think, could be a modality for correcting the frame shift and the nonsense variants. What we need to understand, though, is at the tissue level, what are these changes doing? Because you do need to target your CRISPR construct to specific tissues, and unless if you need to target the central nervous system, the peripheral nervous system, the heart, whichever way, then you kind of need to have some idea of what you're going for and what effect you want to have that's based on the biology of the disorder. Same thing for gene therapy. But if we figure out what the binding partners are, for example, we could look and, downstream, for example, signaling pathways, then we could take a stab at drug repurposing or design of new drugs too. So there's many ways that we can target therapeutics, but they all hinge on what is the function of this gene.
And Loren's absolutely right. One of the things that we had submitted a grant proposal but did not get funded to the NIH was kind of look at the downstream effects of ASXL2. It regulates many other different genes. So we proposed looking in animal and cellular data, looking and seeing what are the downstream effects, and then using currently available FDA-approved drugs to see if we could correct those changes downstream. Unfortunately, the proposal didn't fare well, but that doesn't mean we won't try again. But that's the kind of thing where you don't go directly at the gene change itself, but you look at what happens as a consequence of that change and then try and fix it.
Thank you. Okay, next question is, how do you recommend explaining to other doctors or teachers who want to put Shashi-Pena syndrome kids on the autism spectrum?
I think in order to have autism spectrum disorder, you need to have symptoms of autism spectrum disorder. So, and that's very different from intellectual disabilities or learning disabilities. So I think I don't think you can make a blanket statement that just because you have Shashi-Pena syndrome that you're going to have autism. You need to have symptoms of autism. So it would make more sense to evaluate for cognitive abilities, for example, or learning abilities, and then to make sure that the child is placed in the appropriate type of, and in the least restrictive environment, essentially.
Thank you. All right, and let's see, we'll do the next question is, how old is the oldest person known with Shashi-Pena syndrome?
So we had somebody in their 30s, right? Early 30s. I believe that's the oldest one I have heard about.
Yeah, in our original paper, she was kind of in her mid-30s, and that was in 2016. She's the oldest that we know of. We've heard of a few in their late teens, early adulthood, but nobody else that I know of in their 30s and 40s. That doesn't mean that, they're not out there. They just probably haven't been diagnosed.
I think that there was a question about life expectancy, and
Yes.
I will say that we have not heard of anyone, any deaths that are directly, or any deaths at all, or certainly any deaths due to complications from Shashi-Pena syndrome. So we definitely have not heard about life-limiting complications from the disorder.
And next, we're going to go to a special guest. So Bianca Russell is on with us, and there was a question about the registry and if it's anonymous. And then she also wanted to say a few words about the significance of including more samples from ASXL2 Shashi-Pena syndrome families in the registry. So I'm going to put Bianca on here so she can share a little bit about that. All right, go ahead, Bianca.
Perfect. Thanks for letting me join in for just a couple of minutes. This has been fantastic. And thanks to Dr. Peña and Dr. Shashi, you guys have done an amazing job. I just wanted to give a quick plug for the registry and the biobank as it relates to Shashi-Pena syndrome, because you guys are actually a really important part of the work that we're doing right now. Dr. Wexford did a lovely presentation at one of our national genetics meetings called the American College of Medical Genetics back in March. And she presented some of her work looking at the methylation signature related to ASXL1 and ASXL3. And so that's what Dr. Peña was talking about, how we know these genes are really important in turning on and off certain other genes during development and during someone's lifetime. And we were able to send her one sample from families with ASXL2, and she saw that that person had a very similar pattern to the ASXL1 individuals. And that was really interesting to us, and makes a lot of sense when you listen to what Dr. Peña was saying about how we think ASXL2 works very closely with the other ASXL genes. And there is a lot of really good questions we can answer and explore by looking at how the methylation signature looks in individuals with ASXL2. So it's really, we're launching a pretty hefty recruitment right now to try and collect more samples from ASXL2 families so that we can hopefully help inform more of Dr. Wexberg's work. And also, I think one of the things that was talked about today was this question of what do these missense changes do? And that's something that we can also explore with this methylation signature to see if people with ASXL2 missense changes look similar or different to those with more classic Shashi-Pena syndrome. So I'll just say that please contact us if you're interested in participating. We can certainly explain more. When it comes to anonymity of information within the registry and the biobank, we do find out your information that you share with us through the registry information, but that is kept secure as if it were medical records, and we follow all the HIPAA guidelines that all of your physicians and care providers follow. And then when it comes to sharing that information with other researchers, only de-identified information is shared. So not personal identifying information, but things more like general age, general symptoms, the genetic mutation that you have, but not the details of your name or address or date of birth or anything along those lines. So happy to answer more or take more questions on that, but I think it is certainly an important tool for this community. And hopefully today we've sort of enticed you to learn a little bit more, and please contact us with questions.
Bianca, can you talk about what exactly in terms of sampling is needed if one wants to provide a sample to the biorepository?
Yeah, that's great. Thank you for asking that. We will take whatever we can get. So in the ideal universe, we would love to have blood samples, usually 2 to 3 different tube types, though a small amount, sort of 5 mLs or less in each of those tubes. And then skin samples in a skin biopsy, but we know that those are not always possible. And so it's more common, especially when we're collecting samples from people that we're not seeing in our clinics, to just get a sample of the blood. We are focusing mostly on getting samples from the child or the individual who has Shashi-Pena, but in many circumstances, getting samples from the parents as well can really be helpful, because for a lot of the experiments that people are interested in looking at, having the parents to compare to the child can be really beneficial and serve as a control mechanism. So we certainly will work with you and your family. It depends a bit on where you're located and what we have access to and what works for you and your family. So at minimum, blood samples, but we can certainly talk about collecting more samples if that's something that you're interested in. And I also want to say that Dr. Shashi and Dr. Peña are both a part of the network and a super important part of it. And they are also very much involved in the registry, and you can do sample collection and registry enrollment through both of their practices as well. So,
and let me just put in a plug too. If your child is going for a procedure, like a surgical procedure, you could think of a skin biopsy from one of the incision sites, and that would be kind of a convenient sample and not a dedicated punch biopsy, for example. So I'm putting a plug in, should there be a procedure, a surgical procedure, that is one way to procure a skin sample, a skin biopsy without actually having a biopsy.
Thank you. And if you go, I put in the chat there the link to the registry page on the Our Foundation website. From there, you can access the email address that will connect you to the research coordinator on Dr. Russell's team, and they can help coordinate any of these logistical details there. So if that's of interest to you, definitely reach out to Dr. Russell's team, and they can help make sure that we can get you connected to the resources and information you need to participate in the registry. Great. Thank you, Dr. Russell, for popping in. We appreciate that. All right, so there are no more questions. So I think that brings us to the end here. So I'm going to share my screen one more time here as we wrap up. Okay, so I just want to close first by thanking Dr. Peña and Dr. Shashi for sharing your time and your expertise with us, especially on a weekend. We really appreciate it. And as we close, I just want to invite you to connect with us. We publish a monthly newsletter and announce events like this one through our email list, which you can sign up for on our website. It's a great way to stay tuned for things like the session with Dr. Wexberg, which will be in September. We're also working on a recorded session with Dr. Kadapa, who Dr. Peña mentioned in her slides with that Venn diagram. He's going to do a sort of a genetics 101 for families. So that's upcoming as well. You can follow us on Facebook, on Twitter, and on Instagram. And also just want to invite you to please feel welcome in getting involved with us in advancing research and education by contributing to our work, either as a donor or as a volunteer. And that's my email address there, again, if you'd like to be in touch about any of those opportunities. So finally, just thank you so much for joining us today. You'll receive a link to a survey tomorrow through an automated email from Zoom to share your feedback on today's session. We base our programming on family feedback, so please take a couple of minutes to share your thoughts on today's program, and you can suggest topics for future sessions as well. It's a very brief survey, it's only 3 questions. And finally, at the conclusion of the webinar, you'll be taken to our donate page. If today's session was helpful to you, I invite you to make a contribution to support future programs like this one. So thank you again to Dr. Peña and Dr. Shashi. We appreciate your time, and thanks to everybody for all your wonderful questions. And this recording will be available on the website, hopefully by the end of the day today. So thank you, everybody.
Thank you, everyone. It was a
Thank you.
pleasure. Bye.
Bye.
Bye.